Good evening, everyone. For tonight, I am going to discuss about transcription and RNA processing. For my topic outline, first we have the overview of transcription, second, stages of transcription, and last we have RNA processing. Let us first define what is transcription. Transcription converts a gene into a single-stranded RNA molecule, meaning in transcription, the DNA sequence of a gene is transcribed or copied out to make an RNA molecule. Transcription is also a process in which information is rewritten. Transcription is something we do in our everyday lives, and it is also something our cells must do in a more specialized and narrowly defined way. Transcription is one of the fundamental processes that happens to our genome. It is the process of turning DNA into RNA. To better understand how transcription happens, suppose you want to play skee-ball at an arcade. The game only takes tokens, but you only have quarters. What do? Of course, you exchange your quarters for tokens. In a similar way, your cells cannot make proteins from DNA. They must convert the DNA into RNA in order to make proteins. This process is known as transcription. The central dogma states that information flows in one direction, from DNA to RNA to protein. Transcription makes three types of RNA. First, we have messenger RNA, or the mRNA, which has been transcribed from the DNA and carries the message that will be translated to form a protein. Second, we have ribosomal RNA, or the rRNA, which forms part of ribosomes where proteins are made. And the third one is the transfer RNA, or the tRNA, which brings amino acids from the cytoplasm to a ribosome. RNA is an important type of nucleic acid that plays several roles in the production of protein. RNA is necessary to carry the instructions of the DNA out of the nucleus and to the ribosomes. In transcription, one strand of the DNA that makes up a gene called the non-coding strand acts as a template for the synthesis of a matching RNA strand by an enzyme called RNA polymerase. This RNA strand is the primary transcript. The primary transcript carries the same sequence information as the non-transcribed of DNA, which is sometimes called the coding strand. However, the primary transcript and the coding strand of DNA are not identical. One important difference is that RNA molecules do not include the base thymine. Instead, they have the similar base uracil. Like thymine, uracil pairs with adenine. Again, transcription is the first step in gene expression, in which information from a gene is used to construct a functional product such as protein. The goal of transcription is to make a copy of a gene's DNA sequence. For a protein coding gene, the RNA copy or transcript carries the information needed to build a polypeptide or a protein subunit. Eukaryotic transcripts need to go through some processing steps before translation into proteins. In transcription, a region of DNA opens up. One strand, the template strand, serves as a template for synthesis of a complementary RNA transcript. The other strand, the coding strand, is identical to the RNA transcript in sequence, except that, again, it has uracil base in places of thymine bases. Now let's talk about RNA polymerase. This is the main enzyme involved in transcription, which uses a single-stranded DNA template to synthesize a complementary strand of RNA. Specifically, RNA polymerase builds an RNA strand in the 5' to 3' direction, adding each new nucleotide to the 3' end of the strand. The 5 and the 3 means 5' and 3', which indicates the carbon numbers in the RNA sugar backbone. The 5' carbon has a phosphate group attached to it, and the 3' carbon is a hydroxyl group. Now let's have the stages of transcription. First, we have initiation. In initiation, RNA polymerase binds to a sequence of DNA called the promoter. This promoter turns a gene on or off that is found near the beginning of a gene. Each gene has its own promoter. Once bound, RNA polymerase separates the DNA strands, providing the single-stranded template needed for transcription. The promoter region comes before the transcribed region whose transcription it specifies. It contains recognition site for RNA polymerase or its helper proteins to bind to. The DNA opens up in the promoter region so that RNA polymerase can begin transcription. Now let's have the second stage of transcription, which is elongation. In elongation, one strand of DNA, the template strand, acts as a template for RNA polymerase. As it reads this template one base at a time, the polymerase builds an RNA molecule out of complementary nucleotides, again making a chain that grows from 5' to 3'. The RNA transcript carries the same information as the non-template or the coding strand of DNA. Again, it contains the base uracil instead of thymine. RNA polymerase synthesizes an RNA transcript complementary to the DNA template strand in a 5' to 3' direction. It moves forward along the template strand in the 3' to 5' direction, opening the DNA double helix. As it goes, the synthesized RNA only remains bound to the template strand for a short while, then exits the polymerase. The last stage is the termination. Sequences called terminators signal that the RNA transcript is complete. Once they are transcribed, they cause the transcript to be released from the RNA polymerase. Transcription happens for individual genes, but not all genes are transcribed all the time. Instead, transcription is controlled individually for each gene. Cells carefully regulate transcription, transcribing just the genes whose products are needed at a particular moment. Now let's talk about RNA processing. In humans and other eukaryotes, a freshly made RNA transcript is not quite ready to go. Instead, it's called a pre-mRNA and has to go through some processing steps to become a mature messenger RNA that can be translated into a protein. And this includes addition of cap and tail molecules to the two ends of the transcript. These play a protective role like a book's front and back covers. Second is the removal of chunk sequences called introns. Introns are sort of like blank or messed up pages made during a book's printing, which have to be removed in order for the book to be readable. In this lesson, we'll take a closer look at the cap, tail, and splicing modifications that eukaryotic RNA transcripts receive, seeing how they've carried out and why they are important for making sure we get the right protein from our RNA. Now let's have an overview of pre-mRNA processing in eukaryotes. In this slide, you will see the left panel, which is the eukaryotic cell, and on the right panel is the bacterium. In eukaryotic cell, in the nucleus, a pre-mRNA is produced through transcription of a region of DNA from a linear chromosome. This transcript must undergo processing while it is still in the nucleus in order to become a mature mRNA. The mature mRNA is exported from the nucleus to the cytosol, where it is translated at a ribosome to make a polypeptide. While in the bacterium, the DNA takes the form of a circular chromosome and is located in the cytosol. While the DNA is being transcribed to make an RNA, the RNA there is already considered an mRNA and can associate with a ribosome and start being translated to make a polypeptide. In bacteria, RNA transcripts are ready to act as messenger RNA and get translated into proteins right away. While in eukaryotes, things are a little more complex because the molecule that's directly made by transcription in one of our cells is called a pre-mRNA, reflecting that it needs to go through a few more steps to become an actual messenger RNA. And these steps are: first is the addition of a 5' cap to the beginning of the RNA. Second is the addition of a poly-A tail of A nucleotides to the end of the RNA. And then third is the chopping out of introns or the junk sequences and pasting together of the remaining or the good sequences or the so-called exons. Once it's completed these steps, the RNA is a mature mRNA. It can travel out of the nucleus and be used to make a protein. The first step that pre-mRNA will go through to become an actual messenger RNA is the addition of a 5th cap and a poly-A tail. Both ends of a pre-mRNA are modified by the addition of chemical groups. The group at the beginning of the 5th end is called a cap, while the group at the end is called a tail. Both the cap and the tail protect the transcript. and help it get exported from the nucleus and translated on the ribosomes, which is the protein-making machines found in the cytosol. The fifth cap is added to the first nucleotide in the transcript during transcription. The cap is a modified guanine or the G nucleotide, and it protects the transcript from being broken down. It also helps the ribosome attach to the mRNA and start reading it to make a protein. In this image of a pre-mRNA with a fifth cap and a third poly A tail. The fifth cap is on the fifth end of the pre-mRNA and is a modified guanine or G nucleotide. The poly A tail is on the third end of the pre-mRNA and consists of a long string of adenine or A nucleotide. The second step is the addition of a poly A tail or the tail of A nucleotides or adenine nucleotides to the end of the RNA. How is the poly A tail added? The third end of the RNA forms in kind of a bizarre way. When a sequence called a polyadenylation is signal shows up in an RNA molecule during transcription, an enzyme chops the RNA in two at that site. Another enzyme adds about 100 million, 100,000, 100 up to 200 million, 200,000, 200 adenine nucleotides to the cut end, forming a poly A tail. The tail makes the transcript more stable and helps it get exported from the nucleus to the cytosol. The third big RNA processing event that happens in the cells is RNA splicing. In RNA splicing, specific parts of the pre-mRNA called introns are recognized and removed by a protein and RNA complex called the spliceosome. Introns can be viewed as junk sequences that must be cut out so that the good part of the RNA molecule can be assembled. So what are these good parts? These are the pieces of the RNA that are not chopped out and called the exons. The exons are pasted together by the spliceosome to make the final mature mRNA that is shipped out of the nucleus. In this diagram of the pre-mRNA, which shows the exons and introns, along the length of the mRNA, there is an alternating pattern of exons and introns. Example, exon 1, then intron 1, exon 2, intron 2, exon 3, and so on. Each consists of a stretch of RNA nucleotides. During splicing, the introns are removed from the pre-mRNA and the exons are stuck together to form a mature mRNA that does not contain the intron sequences. Our key point here is that it's only the exons of a gene that encode a protein. Not only do the introns not carry information to build a protein, they actually have to be removed in order for the mRNA to encode a protein with the right sequence. If the spliceosome fails to remove an intron, an mRNA with extra junk in it will be made, and a wrong protein will get produced during translation. Why do we need to splice? Splicing does allow for a process called alternative splicing. in which more than one mRNA can be made from the same gene. Through alternative splicing, we can sneakily encode more different proteins than we have genes in our DNA. In alternative splicing, one pre-mRNA may be spliced in either of two or more than two different ways. For example, in the diagram below, the same pre-mRNA can be spliced in three different ways, depending on which exons are kept. This results in three different mature mRNAs, each of which translates into a protein with a different structure. In summary, transcription is the first step in gene expression, which involves copying a gene's DNA sequence to make an RNA molecule. Transcription is performed by an enzyme called RNA polymerase, which link nucleotides to form an RNA strand using a DNA strand as a template. And then transcription has three stages. First, we have initiation, second, elongation, and the third one is termination. In eukaryotes, RNA molecules must be processed after transcription. They are spliced and have a 5-cap and a poly A tail put on their ends. And also, transcription is controlled separately for each gene in our genome. When an RNA transcript is first made in a eukaryotic cell, it is considered a pre-mRNA and must be processed into a messenger RNA. A fifth cap is added to the beginning of the RNA transcript and a third poly A tail is added to the end. In splicing, some sections of the RNA transcript, which are called the introns, are removed, and the remaining sections, which are the exons, are stuck back together. And then lastly, some genes can be alternatively spliced, leading to the production of different mature mRNA molecules from the same initial transcript. And that's all. Thank you.